all about the central dogma of biology. So we just got done talking about the structure of DNA, and we got talking a little bit about how DNA replicates itself. But now we need to talk about how DNA goes from your cells and makes the traits that you actually have. So, like why you have blue eyes and why some of you are taller and some of you are shorter. All that stuff is coded in your DNA, but how does that DNA actually make the code work? So things you need to know. You need to know what something called RNA is and how it's different from DNA in structure. And you need to know the 3 different types of RNA and how it's involved in something called transcription and translation of your genes. You also need to memorize what this term central dogma actually means. And we'll talk more about it, but the central dogma is just a really simple phrase you have to memorize and understand. DNA codes for RNA, and RNA makes protein. So that's really important. If you don't have that in your notes, write it down right now. We're going to come back to it. The last thing on our objectives is that you have to understand that RNA polymerase is a special enzyme that regulates everything that is happening with RNA and making stuff out of RNA. So there's a lot of, as you can tell, foreign vocabulary. We've talked about this before, about how it's kind of like memorizing Chinese words. You have to memorize and try to understand what all this foreign vocabulary means. So take some time with it and make sure you put the effort into memorizing definitions. Okay, so your genes. We talked about genetics. We talked about how Mendel and his P's figured out that you can predict the different types of outcomes for genes. He didn't know about DNA at the time, but it was discovered, we talked about some of the experiments, it was discovered in the early 1900s, and scientists built the first model, Watson and Crick, 1953, and then it became just this scientific explosion of understanding of what DNA was actually doing. And so our genes that are found on DNA code for certain traits, but we're going to really break down what DNA does. All right, so how exactly does DNA tell our cells what to do and our bodies what to do? And how are these instructions in the DNA made into something that we can actually use? Well, you remember our double helix, and we've got this code in it, right? The A's, the T's, the C's, and the G's. Well, what happens is that that code gives that order, the specific order that those letters are in, gives us instructions for making proteins. And then those proteins do everything. They do all the things in your body. All right, everything that your cells do, all of that is dictated by those proteins that the DNA made. Okay, and so we get our bodies out of those proteins. And there was early confusion in the scientific world about whether the genetic material was actually the proteins themselves, because scientists could tell that they were doing so much. But it turns out that the genetic material was the DNA. Okay. And in fact, this scientific confusion was solved by 2 people named Beadle and Tatum. And I forget their first names. You'll have to read what their first names were. I think one of them was George. But Beadle and Tatum, their experiment had to do with mold and growing mold in a medium, like in a test tube full of nutrients, and they grew some without amino acids and with amino acids, and they exposed these to X-rays to actually mutate the DNA. And they did actually don't expect you to remember all this. I would explain it if we have more time, but we don't. We're getting to the end of the year, right? You can read about this, and what you need to know about all of this mold stuff is that Beadle and Tatum showed that one gene in DNA codes for exactly one enzyme, which, if you remember, is a type of protein. Now, we know that one of these genes now, we've revised what Beadle and Tatum found. One gene codes for what we call now a polypeptide, which is just a piece of a protein or a piece of an enzyme. And then you have to have several polypeptides to make something happen. So anyway, so there's this wonderful scientific experiment. It was pretty awesome. I'm pretty sure they got a Nobel Prize for it, but you can read more about that if you're interested with it. So one gene makes the proteins that do stuff. Now, how does it do that? That's the question we got to address now. And to do that, we have to know about RNA. RNA stands for ribonucleic acid. DNA stood for deoxyribonucleic acid. So you can tell by the names they're pretty similar. The difference is that ribose sugar and a couple other things here. Ribose instead of deoxyribose is a slightly different structure of sugar. It has the same nitrogenous bases except for one difference. Instead of a T, we only have U's. So uracil is substituted in place of all thymine. So there's no T in any of your RNA strands. So everywhere where we would write down those sequences like we did before, instead of putting a T down where A is, you would put a U. Okay, C still goes to G though, so that's good. So we have a slightly different code to work with. Another big difference with RNA is you can see in this picture, this is DNA, this is RNA. RNA is single-stranded, and there are some really good reasons for that, but single-stranded, no T's. Okay, there are a lot of different types of RNA. Okay, we're going to talk about mRNA, tRNA, and rRNA a lot, but there's also siRNA and sRNA and SNRPs, and there's all kinds of things that scientists are learning about RNA. It does a lot of stuff. All right, so quiz yourself here for a second. All right, so what does this all have to do with the central dogma? Well, the central dogma I mentioned has to do with how DNA codes for RNA and that codes for proteins. And we call this the flow of genetic information. All of the code is contained in the DNA, and then it has to get out and be made into stuff. All right? And so how do we move the information from DNA to the rest of our cells and our proteins? Well, this is the flow. The DNA, which replicates itself so that you can get new cells that are exactly the same as your old cells, right? It also, in addition to replicating itself, turns into other things or makes the code for other things, the RNA. And that is a process called transcription. Okay, so this is something you need to know. Transcription means that you go from DNA to RNA. Then RNA takes the information that it got from the DNA and makes it into proteins, and that process also has a name that starts with a T, translation. So one of the things that you'll have to memorize, remember the vocabulary thing, you have to memorize what the differences between transcription and translation is. DNA to RNA is the transcription part, RNA to protein is the translation part. And then we get our traits. So in reality, it's kind of funny that we talk so much about DNA, but it's really a pretty big process, and proteins are really doing all of the work to make our traits when you come right down to it. All right, here's another representation of this, thinking about our DNA as being a code that's locked up in a safe, kind of like a blueprint for making a building or something. We keep that DNA locked up tight inside the nucleus. Okay, but we still have to access it, and it still has to be used for stuff. So in order to do that, we have this tool called mRNA that inside the nucleus of your cell is going to go ahead and make copies that are then distributed to all the different builders and construction workers that need them. And so the mRNA, through transcription, is created. And then through the translation part, the proteins then actually take those plans and make stuff that is useful. And they do it outside of the nucleus. So the RNA leaves the nucleus and goes out into the cytoplasm of the cell where it makes all of these proteins. Okay, and if you remember from way back in chapter 6, Proteins are made of chains of amino acids, which will become important here in a little while. Okay, so that's translation. And traits. So, 3 types of RNA you've got to memorize. The big important players. Messenger RNA is structured as a long strand of nucleotides, single-stranded, remember, that are basically a one-to-one copy of those DNA bases, except for the uracil thing. Second type, rRNA, ribosomal RNA. It's made actually in the nucleolus, partly. The ribosomes kind of get constructed, and these are really important pieces of RNA that are going to do the work of putting amino acids together in long strands of proteins. Okay, and then tRNAs. They transport amino acids around, so they're easy to remember because tRNAs like transfer or transport amino acids to the ribosome. So they're kind of hauling things around. Those are the 3 types, and here's what they look like. Long strand, mRNA. rRNA, this is a ribosome, and this is really, really important for you to remember that rRNA is a fancy word for ribosome, and if you remember back from chapter 7 or chapter 8, whenever we memorized all those things, that's what a ribosome looks like. It's made of rRNA. And then the tRNA, they're kind of funky looking shapes. I always kind of think they look a little bit like a trumpet, but I don't know, maybe. All right, so getting right down into the nitty-gritty here. Transcription. When we transfer the code from DNA to mRNA, we have to go through, and this enzyme is doing all of the heavy lifting, the enzyme called RNA polymerase. DNA gets unzipped, all right? So we pull apart the strands of DNA from each other, and RNA polymerase, this green enzyme right here, goes in and binds to the section where the mRNA is going to be made. In other words, it's going to figure out what gene it needs on our DNA, and it's going to go in and copy just that part of the gene. And so it comes in here at lightning speed, and it just adds all these little bases on. Boom, 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 boom. And it's going to go in here, and this little ribbon of mRNA is going to come out of it. All right, and so the transcription, the important piece to remember here is that wherever you have a G, you're going to get a C. Wherever you have an A, you're going to get a U. G, C, T on the DNA strand will end up being A. So you need to know how those pair up. Okay. Now another interesting thing about this is that the code on the DNA is not all useful. Some of it is periodically interrupted by things that are not in your, they're actually not in your genes. So we have what are called introns and exons. Okay, pieces of DNA that are both useful and not useful. And the not useful parts are called introns. They get taken out during a part called RNA processing. And the useful pieces are left in, and they're called exons. Now this is totally backwards from how you usually think about it. Introns get taken out, exons get kept in. All right, so just memorize that backwards. Introns are non-coding or the in-between sequences, and exons are the expressed sequence, so they get to stay in there. Okay? All right, so we make RNA, it gets processed, then it leaves the nucleus, and it's going to go find the rRNA that was already made, and it's all floating out there in the cytoplasm or attached to the endoplasmic reticulum. And in the process that it finds these, it's going to run through these and make the long chains of amino acids. So it goes out of the nucleus, and this process, the next part, the translation part, happens now. When it finds the ribosome, which is made of 2 little pieces here, it's going to bring in these tRNAs that have an amino acid. Remember, they carry around the amino acids. And this mRNA is going to come inside of the rRNA or the ribosome. So this is the mRNA, this is the rRNA, these are the tRNAs. And based on what the code is, or the order of these is, it's going to attach a different amino acid. So if it has an A, a U, and a G, it's going to attach a different amino acid based on whatever order they're in. Okay, so those are really important pieces. The tRNA, the amino acids, long chains of amino acids, which are polypeptides. Okay, so lots of new stuff going on here. Now, the codes, the order, these little bases are divided up into groups of 3. Okay, and experiments in about the 60s demonstrated that the code was based on 3s. Why it's based on 3s, not 4s? Well, it could have been 4, but it's not, it's 3. So these 3 bases together are called a codon. And the codons form this language of protein making. It's really, really amazing. All right, so these 3 codons, in whatever order they're in, are going to determine what your protein is going to look like. Okay. Now, in the translation part of it, we mentioned the tRNA a couple of times. It actually has the job of interpreting this codon sequence. So your mRNA chain, say you have, your A, your G, your C, whatever it is, and you come to a specific 3-letter code, the tRNA will read it, and it will decide which amino acid you need to take on. And so if it's UGG, for example, it will have this little matching piece that will tell it, oh, you need tryptophan, and so it will go to stick on a tryptophan amino acid. Okay, this little piece on the tRNA is called an anticodon. And so the anticodons and the codons match up and tells you what amino acid you need to stick onto the process. Okay, now this is kind of a demonstration of how this looks overall, and you need to really understand this, so pause if you need to and process it. But the mRNA is going to be coding for proteins, and it looks like this. You might have a long strand of DNA of this sequence. And once you go through in the transcription process, you end up getting something that looks like this. Remember, your A's will go to U's instead of to your T's. And so here's your mRNA. And then you run it through the ribosome outside the nucleus, and then suddenly you have these, which are amino acids. That stands for methionine, arginine, valine, and so on. And all of these are different, and the order dictates what protein you make. Okay? And so these are in groups of 3s here that directly correspond to these amino acids. All right? So the codons are very, very important, and they will tell you what amino acids you put in. Now there's a chart. There are really only so many codons that are possible. You only have 4 letters to work with. And so you can figure out how many different combinations of letters are possible. It's 60 something. But some of those are duplicated, and so the same 3-letter codon will actually make the same amino acid. So if you have the sequence CCU and CCC and CCA, you'll make proline for all of those. And so there's a lot of duplication here. And that's probably a good thing, because if it were too specific, we'd just have kind of a mess. But that's, that's a whole different subject. But this is the code for every single living thing on Earth. Now that's pretty cool when you think about it. The chemistry didn't have to come out this way. It doesn't have to be so specific and so orderly, but it is. And every living thing shares this code. There is no other code for life on Earth. And it's one of the strongest supports that we have for the origin of life being a common ancestor, is that we are all made of exactly the same stuff, exactly the same code, exactly the same DNA. All right, the code is also very redundant, and that's what I was saying about how you have your different codons that code for the same thing. It's kind of a safeguard so that we don't have mistakes, but several codons will code for each amino acid. There's also one codon that is what we call the start codon, and that's how an mRNA sequence knows where to start making a protein. And the start codon is this one right here, AUG. So anytime you have an AUG, that is the signal, okay, start right here and make your protein. And so it will be the place where that signal to start. And then there are 3 different stop codons. Okay, the stop codons can either be UAA, UAG, or UGA, and that's the signal to, okay, stop making your protein right here. So that's all pretty cool stuff, really pretty miraculous. Okay, now at this point you should pause the video and think about this picture here. See if you can describe what's going on in here. All right, I'll just assume that you did that and thought about it for a second, and I want to tell you about it here, but I'm going to use the next slide. Nope, I'm not. The DNA here is inside of the nucleus. That's the big purple thing. It unzips. mRNA is made. It's processed with the introns being taken out, leaves the nucleus through the nuclear pores, finds the ribosome. These represent tRNAs right here, where the anticodon of the RNA, the tRNA, is meeting up with the codon of the mRNA, figuring out which correct amino acid is going to come in, and it makes the big long chain. And that goes on and does whatever specific job it's designed to do so that you can have a trait and have, you know, the right chemicals in your body and the right size and shape of all your organs and the right functions, and everything that happens in your body is due to those proteins. All right, this is the picture in your book. It's really similar to what I just showed you, so I'm not going to go through it again, but you need to know this whole process, and it's tricky. It will require you to study it and think about it really carefully, and maybe even watch the video again. All right, so that's the end of it. Hope you have a wonderful Monday. We'll see you tomorrow.